So in this video, we're going to talk about antimicrobial drugs, or more specifically, how bacteria become resistant to antimicrobial drugs. And so there are four main mechanisms for bacterial antibiotic resistance, meaning ways that bacteria have evolved to make them less sensitive to a particular antibiotic. And the first mechanism for this is going to be reduced permeability. And what this means is basically that in this case, the antibiotic is less likely to get in. And this happens through several mechanisms. It could be that you get decreased expressions of these porins. And remember that porins are found in gram-negative bacteria in their outer membrane. And it might be that possibly the bacteria stop, I should say, producing the porins at such a high level. And because of the lack of porins, the antibiotics are less likely to get in. Another possibility for reduced permeability is that there could be a physical change in the porin protein to reduce the permeability, meaning that they get a mutation in the porin that makes the antibiotic less likely to get in. And lastly, this could be a change to the cell wall structure. And that can happen because maybe the bacteria acquire the ability to produce capsules or slimes. And remember that capsules and slimes help to protect the bacteria against antimicrobial control mechanisms. The second mechanism for antibiotic resistance is restricted access of antibiotics, meaning that in this case, although the antibiotic might get in, there are these efflux pumps, and these efflux pumps are basically these pumps, and this is the bacteria's way of pumping the antibiotic back out of the cell, meaning the antibiotic gets in, but now it's going to pump it back out. Think of it, it's kind of like it's a bouncer at a club. It's going to push it back out and say, you can't come in here. The third type of mechanisms for antibiotic resistance are altered targets of the antibiotic. And so here we go. And in this case, this prevents the antibiotic from binding to the target molecule caused by the mutation. And this can be seen for almost every antibiotic class. An example of this is, remember that we talked about that bacteria have an enzyme that converts PABA to folic acid. And remember that sulfanilamide, or the sulfa class of antibiotics, act as competitive inhibitors to PABA. They act as competitive inhibitors for the enzyme that converts PABA to folic acid. And so in this case, the enzyme itself might get a mutation so that sulfanilamide can no longer bind and no longer inhibits folic acid synthesis. And so that's what we mean by we say antibiotic target site alteration. So that enzyme changes, that's the target site, and because of that, sulfanilamide no longer binds, and it's no longer able to prevent the bacteria from producing folic acid. The last one is going to be antibiotic inactivation. And this can occur either through the degradation of the antibiotic, meaning that they might actually break down the antibiotic, or potentially the antibiotic becomes modified. And this modification might prevent the antibiotic from binding to its target. For example, bacteria like Staphylococcus, for example, remember, can produce beta-lactamases, or more specifically penicillinases, and that those enzymes are able to inactivate beta-lactam antibiotics, meaning that they cut that beta-lactam ring, and that no longer allows the penicillins or the cephalosporins from being able to inhibit cell wall synthesis. And so now we're going to look at, how do bacteria come to be antibiotic resistant? Like, how do they gain the ability to do these various things? So let's look at how bacteria acquire the ability to become antibiotic resistant. And the first is what we call vertical gene transfer, and that occurs during reproduction between generations, meaning going from one generation to the next. And so in humans and plants, this happens going from parents to their offspring. In bacteria, remember that bacteria don't reproduce sexually. Instead, they reproduce asexually, which simply means that they just make a copy of themselves. And they do that through DNA replication, meaning that they're going to make an exact copy of their DNA to produce two daughter cells. However, the enzyme responsible for this is DNA polymerase, and DNA polymerase is not perfect. DNA polymerase during replication can make mistakes, and if it makes a mistake, it's called a mutation. And so a mutation can be a way that through vertical gene transfer, meaning again we're going from parent to offspring, and that mutation might make it so that they acquire the ability to be antibiotic resistant. For example, this could lead to a mutated porin. Remember in the last slide, porins can become mutated and make it less likely for the antibiotic to get in. This can also lead to altered antibiotic target sites. So again, like the example when we talked about PABA being used to make folic acid, it might be that you get a mutation in that enzyme that no longer makes sulfanilamide able to inhibit folic acid synthesis. And so this would be considered vertical gene transfer. The next one is what we call horizontal gene transfer. And this is the transfer of genes within the same generation. And so there are three main types of horizontal gene transfer that we will look at. The first is going to be transformation. The second will be transduction. And the third is going to be conjugation. And in the next slide, we'll talk more specifically about this. Now, in order for there to be horizontal gene transfer, again, it's within the same generation, and this is basically a way that bacteria can transfer antibiotic resistance genes from one cell to another. And these are typically going to be transferred on what we call plasmids, and you learned in lab that plasmids are extrachromosomal DNA sequences. And when we get to chapter 8, we'll talk more about what are called transposons, which are these jumping genes, and that these can be passed from one organism to another. And because of these resistance genes being able to be transferred, this can lead to what we typically call superbugs, which are bacteria that are resistant to a large number of antibiotics. They might be resistant to only one if they're not a superbug, or in some cases, some bacteria are resistant to many types of antibiotics, and those would be the ones that we would refer to as these superbugs. So now we're going to talk briefly about the three types of horizontal gene transfer, and we will talk more about the specifics of this when we get to chapter 8. And so the first mechanism for how bacteria acquire the genes necessary for antibiotic resistance is through what we call transformation. And this is when naked DNA is transferred from a dead donor into the competent recipient. And so if we look, here we have our dead donor cell. And when this donor cell dies, it lyses open and it might release some of its genetic information. And so if you look here, notice, so here are these DNA sequences, and one of these might be an antibiotic resistance gene. Remember in lab, when we looked at transformation, we treated the recipient cells with calcium chloride. That neutralized the charges on the DNA, and that allowed the bacteria to take up those DNA sequences. It wasn't from a living cell, it was from DNA that was simply outside the cell, and the DNA went in, and if it incorporated into the recipient cell, now the recipient cell has acquired the ability to become antibiotic resistant. The next mechanism is something called transduction. And transduction uses a virus, and specifically what we call a bacteriophage, which is just another name for a type of virus that infects bacteria. And the phage acts as a genetic vector, passing DNA from the donor to the recipient. And if that donor DNA incorporates into the recipient, that then allows the recipient cell to become antibiotic resistant. Remember in lab, when we set up a plaque assay, we used the T4 phage, and the T4 phage infected the E. coli. Now, during this process, it's totally possible for the bacteria to pick up some of the donor DNA before the virus causes the bacteria to lyse. So if you notice, here is this phage-infected donor cell, and it's possible that during phage replication and production of new phage, that it might incorporate that antibiotic resistance gene into the phage genome. And then when that donor cell lyses and it releases the phage, and the phage go on to infect a new cell, now when they inject their DNA into the recipient cell to make this cell be a virus-producing factory, it's possible that this antibiotic resistance gene incorporates into the recipient cell chromosome. And now that makes this recipient cell also able to be antibiotic resistant. And the last mechanism is what we call conjugation. And conjugation is going to be the transfer of genetic material from one cell to another involving cell-cell contact. So notice in the first two mechanisms, it's not that the cells come in contact. In the first one, the cell takes up DNA from the outside. The second mechanism, it was through a phage. The phage acted as a vector. The third one, though, when we talk about our conjugation, now we're getting cell-cell contact. And so, in gram-negative bacteria, there are plasmids that carry genes that code for the formation of sex pili. And remember that sex pili are projections from the donor cell that contacts a recipient and helps to bring the two cells into direct contact. And so during conjugation, when we get this plasmid allows the bacteria to produce the sex pili, and then during replication of this plasmid, it might transfer some of that plasmid into the recipient cell. And now the recipient cell has the antibiotic resistance gene, and it might also have the gene that allows this recipient cell to produce sex pili as well and keep passing on genetic information. Most of you have probably heard about MRSA in the news before. And MRSA stands for methicillin-resistant Staphylococcus aureus, and again we pronounce that MRSA. And this essentially means that this strand of bacteria is resistant to many of the antibiotics that we have to treat this type of infection. This is the leading cause of healthcare associated infections, meaning that you acquire this infection while in a healthcare facility being treated for something else. According to the Center for Disease Control, the CDC, in 2011, there were 461 cases of MRSA reported, and in 11,285 of those cases resulted in the death of the patient. The good news is that MRSA cases are starting to decrease in the healthcare setting. The bad news is that there have been 13 cases of what is called VRSA, which stands for vancomycin-resistant Staph aureus, since 2002. And vancomycin is generally considered to be a last resort antibiotic for patients because it has high toxicity. However, some strains of Staph aureus are now resistant to our last resort antibiotic, and that's a scary thought. So, studies show that about 1 in 3 individuals carry Staph aureus, shown here as these grape-like clusters, in their nose and on their skin, usually without illness. About 2 in 100 individuals carry MRSA, and MRSA is typically spread from person to person on contaminated hands, skin, or objects. In the community, most MRSA infections are skin infections, like the one shown in the picture, and it starts out as a small red area, and it might turn into a painful red, swollen, warm to the touch abscess that fills with pus in a matter of days. One of the ways that doctors can determine if the abscess is caused by Staph aureus is to swab the wound and then grow it on mannitol salt agar like we did in lab. The special plate, the MSA agar, is formulated to grow Staph aureus taken from the skin. If you see growth on an MSA plate and it turns the agar yellow, then it's probably Staph aureus because Staph is osmotolerant, meaning it can grow on high salt, and it can ferment the mannitol to produce acids, which causes the fennel red to turn yellow. And so you guys got to test yourself to see if in fact you carry Staph aureus as part of your normal flora. Now, that's to tell you if Staph aureus is present. If we want to look at if the bacteria is antibiotic resistant, like to be MRSA, the wound is swabbed, and then an antibiotic sensitivity test will be performed like we did in the lab. Remember, we did our Kirby-Bauer test. If the bacteria grows right up next to the antibiotic discs, so as you can see here, as the bacteria grew right up to the disc, that tells us that the bacteria is resistant to the antibiotic, meaning that the antibiotic had no effect on the bacterial growth at all. However, if there's no growth around the antibiotic disc, then we would say that the bacteria is sensitive to the antibiotic. And so notice here, we have this clearing around the disc, which means that in this case, this antibiotic on this disc inhibited bacterial growth. Notice that if we look at the plate for MRSA, there's only one antibiotic that the MRSA is sensitive to, so notice here, and that is the vancomycin. Now, notice that the growth on the plate is gold color, and its golden color is where the species name comes from. Remember, aureus, AU, because AU is the atomic symbol for gold. Now, although in the community, MRSA only causes typically a skin infection, in medical facilities, MRSA can cause life-threatening bloodstream infections or sepsis. It can cause pneumonia, necrotizing fasciitis, which is flesh-eating disease, as well as surgical site infections. And so in this case, MRSA can be actually quite dangerous. So this is a question to get you guys thinking. In addition to not finishing a cycle of antibiotics or taking it as prescribed, what else can society do to minimize the creation of new antibiotic-resistant bacteria? And so remember, we did the example in class about how when you don't finish an antibiotic, like let's say you were prescribed a 10-day dose, if you only take that antibiotic for, let's say, 3 days, well, then now you're killing off the weaker bacteria first and allowing the stronger bacteria to survive, and you end up with a population that is more antibiotic resistant than the initial population. And so we want to think about what else can society do to try and minimize the creation of new antibiotic resistant bacteria. And so I want you to just go ahead and pause the video, think about your answers, and then we will go ahead and talk about what these ways are. So when you're ready, go ahead and push play. So the first one that we will talk about is that one of the ways that we can prevent antibiotic-resistant infections would be to prevent the infection in the first place. And so how do we prevent the infection in the first place? Well, this can occur through hand washing, proper food preparation, meaning to follow proper food safety, immunizations if possible. And so if we don't get those infections, then they're not likely to become antibiotic resistant bacteria. The second way that we can prevent this is to prevent the spread of the infection. And so, again, like in a healthcare setting, taking proper precautions to try and prevent the spread of that infection from one patient to another. And so that would be one way that we can help to minimize the creation of new antibiotic resistant. A third way is to track infections. And what that means is that the CDC, or the Center for Disease Control, begins to track these infections to learn how they spread, the risk factors for contracting the disease, etc. Again, to better study how to prevent the spread, like how do we prevent passing this from one patient to another? And that then allows us to take proper precautions. The next one is to improve antibiotic administration and stewardship. And what that means is basically this would be to help basically get doctors to not prescribe an antibiotic that is not needed. Let's say, for example, a patient has a cold. Remember that the cold is caused by a virus. Antibiotics are not effective against a virus. And so in some cases, patients will go to the doctor because they're not feeling well, and they might demand that the doctor give them an antibiotic so that they can feel better. And in some cases, it might be that the doctor feels this pressure, and just to get the patient to stop, they might actually prescribe an antibiotic when they know that it's not effective. And so it would be important to basically help to get the word out to physicians to really be careful with the way that they prescribe antibiotics. That antibiotics should only be prescribed when necessary, meaning that the patient has a bacterial infection. And so basically improving the way that antibiotics are administered. The next one is going to be to stop using antibiotics in livestock. And so in some cases, farmers have been giving livestock, like cattle, for example, antibiotics, and the antibiotics help to protect them against bacterial infections, and it might actually allow the animal to become bigger in size in some cases, which produces more meat. And so a lot of times farmers will give preventative antibiotics to livestock to basically help with this. Now, the problem with this is, again, the more often you give the antibiotics to these animals, you also might lead to more antibiotic resistant bacteria. And then the last one would be develop new antibiotics and tests, so that we have more antibiotics that can be used in cases where our bacteria become resistant to all the current antibiotics that we have. So, question for you. If one measures a large zone of inhibition in the disk diffusion test, one can assume that the bacteria are red, sensitive to the antibiotic, yellow, resistant to the antibiotic, green, unaffected by the antibiotic, or blue, are producing the antibiotic. So go ahead and pause, think about your answer, and then come back and turn on the video. So if you said red, that the bacteria are sensitive to the antibiotic, that is true. If you see that large zone of inhibition, remember that means that there's a big clearing around that disc. And if there's big clearing, that would tell you that the bacteria is sensitive, meaning that the antibiotic is effective to inhibiting the bacteria's growth. And so that concludes our video.